Thank you, Brenda, for your kind introduction. I'm happy to be here this afternoon to make this presentation to you. Before I get started, I want to re-emphasize some of our quality position for OPQ. Pharmaceutical quality, a quality product of any kind, consistently meets the expectations of the user, and drugs are no different. Patients expect safe and effective medicine with every dose that they take. And pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It's what gives patients confidence in their next dose of medicine. So along those lines, I'd like to present to you today the current agency thinking on conducting test and reference product comparability studies for complex peptide ANDAs. Okay, so I'll start with an outline of the presentation. I'll first go over the regulatory pathway for synthetic and generic peptides. I'll then discuss the concept of therapeutic equivalence in the context of peptide applications. And then I'll speak briefly about peptide characterization at the API level. And the bulk of my presentation will be regarding peptide comparability studies, which will be to be conducted in comparison between your proposed product and RLD at the drug product level. And these studies include higher order structure, aggregation profile, impurity profile, and biological activity. I'll also discuss the consequences of allowable formulation changes under the regulations on conducting these comparability studies. And then I'll finish with some discussion of the container closure system, relevant issues there. Okay, so first the regulatory pathway. The agency considers a polymer of 40 or fewer amino acids to be a peptide regulated under the Food, Drug, and Cosmetics Act. And so in recent years, a pathway for the development of complex peptide drug products under Section 505J has been facilitated by a number of factors. And these include, firstly, the development of solid phase peptide synthesis, and also the development of highly selective and sensitive analytical techniques. So these technologies have really facilitated the potential to assess immunogenicity for these types of peptide products manufactured using these procedures. Because peptides manufactured this way have defined starting materials, they have characterizable peptide-related impurities, they generally have no glycosylation, and they don't have any host cell proteins that are typical contaminants of peptides manufactured by recombinant pathways. So along those lines, a draft guidance for industry was published in draft form October 2017 regarding certain highly purified synthetic peptide drug products that reference RLDs of recombinant DNA origin. As Dei mentioned, there are 5 peptides that are covered by this guidance, but for any other peptide products that aren't covered, the immunogenicity risk, and hence their eligibility for approval under Section 505J, is assessed on a case-by-case basis. Okay, so let's discuss therapeutic equivalence. For any ANDA that's submitted under Section 505J, applicants must demonstrate that their proposed product has therapeutic equivalence with the RLD. So in order to demonstrate therapeutic equivalence, you must demonstrate both pharmaceutical equivalence and bioequivalence. Pharmaceutical equivalence, to demonstrate this, you must show that your proposed product has the same active ingredient or ingredients, is of the same dosage form, delivered by the same route of administration, and is of the same strength. For bioequivalence, it stated under Section 21 CFR 320.22 that a drug product's in vivo bioavailability or bioequivalence may be considered to be self-evident for drug products that are parenteral solutions that are intended for administration by injection, and also contain the same active ingredient and inactive ingredients in the same concentration as the RLD. So in other words, the concept of that is the Q1, Q2 comparison. Now, most peptide products tend to be injectable products, and hence they tend to be eligible for a bio waiver. So the overall mitigation of this risk of demonstration of therapeutic equivalence really comes down to demonstration that your proposed product has the same active ingredient or ingredients. So along those lines, we asked firms to provide comprehensive characterization of both the peptide structure and various other aspects, along with a series of comparability studies. So at the API level, we asked that applicants provide evidence to ensure the identity, strength, quality, and purity of their peptide drug substance. And this, of course, includes the characterization of the primary peptide structure, which means the amino acid sequence and any side chain modifications that may be present in the API of interest. And there's also a series of physicochemical properties which Dei summarized in his presentation. Now, these studies are often provided by the DMF holder in the instance where a DMF is referenced for the manufacture of the API. But we also ask that ANDA applicants provide this data one time in-house to facilitate product understanding. Now, at the drug product level, we ask that you provide evidence to ensure your peptide higher order structure, aggregation profile, impurity profile, and biological activity are comparable to those of the reference product. And these studies should be conducted in the fully formulated drug product, so not just on the purified API. The studies are not only important for demonstrating that your proposed product has the appropriate biological activity, but they're also an integral part of assessing the overall immunogenicity risk of your product. Okay, so let's just discuss in a little more detail the various types of comparability studies that we expect. So first we have higher order structure. Now, higher order structure consists of both secondary and tertiary structure. And secondary structure consists of structural features such as alpha helix, beta sheet, random coil, or unordered structure. And tertiary structure refers to the overall monomer structure, or essentially the three-dimensional structure in solution. Now, for aggregation, aggregates can range in size from small oligomers, for example, soluble dimers, all the way up to large assemblies that exist in the subvisible to visible spectrum. They can form during production, storage, shipment, or delivery. And they can also form by nucleation around foreign particles that may be contaminants in your proposed product. For example, steel or rubber particles that may originate from the container closure system or manufacturing equipment. For impurities, the comparability study here is generally useful for mitigation of the risk of process impurities, because one would expect that any degradative impurities that are present in your proposed product would be the same as they would be for the RLD, of course, assuming that the identification of your API is the same. And finally, for bioassay, we ask that firms provide a comparative study of the biological activity of your proposed product using a biologically relevant assay, and justification should be provided for the choice of that assay. Okay, so before we move on to some more detail about each study class, I want to discuss some general considerations for drug product comparability studies. First, I mentioned earlier that peptide drug products tend to be parenteral products, that is, they're either sterilized solutions or lyophilized products to be reconstituted for injection. So along those lines, we ask that you conduct your comparability studies on the finished drug product where possible. And that includes reconstitution per the label instructions if necessary. Now, if it's not feasible to conduct a study on the finished drug product, we ask that you provide justification for the reasoning behind that. And that could be because of a limitation of an analytical technique or some sort of interfering component in the formulation. But in any event, we ask that you endeavor to keep sample manipulation to a minimum when you're designing these studies. So each study should be conducted on a statistically meaningful number of lots of both your proposed product and the RLD, both tested on in the release and at or near the end of the proposed shelf life or expiry date. And samples should be aged under conditions consistent with the label storage conditions. We ask that you use multiple orthogonal validated methods for each comparability study class, and that you provide sample dates for the sample ages for the dates of all studies. And finally, we ask that you provide the drug substance lot number that's used for manufacture of each drug product lot that's used in these studies. Okay, so let's discuss higher order structure in a little more detail. So these comparability studies, the goal of them is to be able to demonstrate that the higher order structure found in your proposed product is comparable to that of the reference product. I'm going to list some examples of various methods that can be used for this characterization. By no means am I intending to prescribe a technique that's general for all peptides, but you should provide justification for the choice of the methods you use for this characterization as they are relevant to the API present in your proposed product. But for characterization of secondary structure, we do see the use of far UV circular dichroism, FTIR and Raman spectroscopy. For characterization of tertiary structure, we see the use of near UV, CD, intrinsic fluorescence, and differential scanning calorimetry. And for characterization of tertiary structure, NMR spectroscopy is probably the most powerful technique. And so for drug products that contain significant levels of high order structure, we would likely expect that NMR spectroscopy would be one of the choices, one of the methods chosen. Okay, so for aggregation comparability studies, the goal here is to demonstrate that the levels of aggregation found in your proposed product are the same as or less than those of the reference product. So for these studies, in addition to aging samples under label storage conditions, we also ask that you conduct these comparability studies on samples that have been subjected to stress conditions. And that stress conditions could include exposure to elevated temperature or exposure to light. Again, I've listed some examples of methods here, but you should provide justification for the choice of your methods as they're relevant to the API present in your proposed product. So light scattering is a detection method that's commonly used for detecting aggregates. It can be coupled with size exclusion chromatography in the form of multi-angle light scattering. We also see the use of composition gradient multi-angle light scattering and dynamic light scattering. Analytical ultracentrifugation is often used, and we see the use of both sedimentation equilibrium and sedimentation velocity, AUC. We often see the use of gel electrophoresis, both under denaturing, that is the standard SDS page conditions, or also the use of non-denaturing or native conditions. And we sometimes see the use of capillary electrophoresis, which is a related technique. Field flow fractionation is often used, usually in the modern guise of asymmetric field flow fractionation. And then finally, microflow imaging is more of an optical technique, which is useful for characterization of aggregates in the subvisible to visible spectrum. Okay, so before I leave high order structure and aggregation comparability studies, I want to discuss briefly allowable formulation changes that are allowed under the regulations. So specifically in 21 CFR 314.94, the various inactive ingredient changes that are permitted for ANDA drug products is discussed, or ANDA drug products for parenteral use. Specifically, what it says here is that an applicant may seek approval of a drug product that differs from the reference listed drug in preservative, buffer, or antioxidant. But under these, if such a proposal is made, the applicant must identify and characterize the differences and then provide justification demonstrating that the differences do not affect the safety of or efficacy of the proposed product. So how does this affect the comparability studies that you've provided for your application? So we ask that the risk of the changes such as these be mitigated by further studies of higher order structure and aggregation in comparison to the reference product. So for example, if you're proposing a change to the buffer, which is a change that we sometimes see for peptide applications, the risk here is that deletion of or changes to the RLD buffer could potentially result in fluctuations of the pH outside of your proposed pH range and or changes to the peptide conformation in solution and aggregation levels. So what we ask in this situation is that you provide additional comparative higher order structure and aggregation studies conducted on samples whose pH has been adjusted across your proposed pH range and slightly outside of those pH range in comparison to the RLD in its marketed formulation. And this is just to ensure that the different buffer does not change any of these critical attributes. Okay, so let's move on to a discussion of peptide impurities. Impurities and degradants may form during manufacture and storage. They may result from the insertion, deletion, or modification of amino acid sequences or residues. They can be process in nature or degradative, or they could be both. So here is a list of various mechanisms of formation of peptide impurities. And those at the top of the list tend to be impurities that are formed under degradative conditions, and those at the bottom tend to be impurities formed under manufacturing process type conditions. In the middle of the list, these mechanisms can form under both sets of conditions, either during manufacture or on storage. Okay, so for impurity comparability studies, what do we expect here? So We ask that you provide a profile of the impurities in your proposed product in comparison to the reference product. Firstly, so you can demonstrate the impurities common to both your proposed product and the RLD are present in your proposed product at the same or lower levels in the RLD. So this is the standard of quality that you should be driving towards. Now, for any new impurities that are present in your proposed product that are not common to the reference product, we ask that you fully characterize these impurities. Again, the study should be conducted on a statistically meaningful number of lots of your proposed product and RLD, tested across the shelf life, and the sample should be aged under conditions consistent with label storage conditions. We ask that you use multiple orthogonal validated analytical methods, and you should consider the use of UHPLC high-res MSMS, not only to facilitate peak identification, but also ensure peak purity, in other words, the specificity of your method. Provided a link or an agency reference here where this technique is used. And also DE's presentation previously also described the use of this technique for characterization of impurity profiles. I want to briefly mention impurity limits. So ICH Q3A and B, both these guidelines state that they don't apply to peptide drug products. However, they can be used as a starting point for establishment of peptide impurity limits. However, ultimately, we ask that your proposed limits be justified by levels observed in the RLD or in the absence of this type of data by safety evaluation, and that could include evaluation of toxicology and immunogenicity as appropriate. Getting back to the peptide guidance that we mentioned earlier in the presentation, again, it was published in 2017 and covers 5 peptide drug products: glucagon, liraglutide, xerotide, teriparatide, and teduglutide. And in this guidance, what is required to submit an ANDA via the 505J pathway for these 5 products is described. So first, you must identify each peptide-related impurity present in your proposed product at a level of 0.10% of the drug substance or greater. So this is where the analytical methods are crucial, the choice of these methods to facilitate the identification of all of these peptide impurities. For each peptide-related impurity that's done in your proposed product and the RLD, the proposed product level of such an impurity should be the same as or lower than that found in the reference product. So that's what I mentioned on the previous slide, and that still applies here. It's the standard of quality that we're shooting for. You should also be able to demonstrate that your proposed product does not contain any new peptide-related impurities at levels greater than 0.5% of the drug substance. So if you cannot demonstrate this for your particular product, the 505J pathway is not an option. You are disqualified from that pathway. So you should be striving to meet that goal, that particular point as well. And then for any peptide-related impurities that are found between 0.1 and 0.5% of the drug substance that are either not present in the RLD or are present in the proposed product at levels higher than the RLD, you must provide justification as to why these impurities don't affect the safety or efficacy or potential for immunogenicity of your proposed product. So along those lines, for these types of impurities down between 0.1 and 0.5%, you must provide an evaluation of the risk of immunogenicity. And this includes evaluation of each individual impurity of interest for its potential to activate T cells via binding of the peptide-related, by binding of these peptide-related impurities to the major histocompatibility complex. This includes in silico studies of MHC binding and in vitro binding and functional assays of those specific purified impurities. We also ask for a comparison of innate immune activity that's conducted on your proposed generic formulated product against the RLD, and that can be done using both in vitro cell-based assays and animal models. Okay, so the container closure system, you should evaluate your proposed container closure system with respect for its ability to protect your proposed product from external influences such as exposure to light, its overall compatibility with the drug product solution, and safety and performance. We expect that you perform both extractables and leachable studies using sensitive analytical methods such as GCMS or HPLC MS. And then, but really the design of your container closure system should be such that it's compatible with the drug product to minimize the amount of leachables. So we ask that you demonstrate the absence of significant levels of leachables in your proposed product, if you're unable to do this, you can demonstrate comparability of the leachables profiles between your proposed generic product and multiple batches of the ALD over the product shelf life. Okay, so I'll just finish by summarizing the presentation. We discussed the various comparability studies that should be conducted and provided in your application between the proposed product and ALD. And we discussed how the following should be provided: high order structure, aggregation profile, impurity profile, biological activity, and container closure system. We also discussed the allowable formulation changes that will likely require further comparability studies. So I will finish there, and thank you for your attention, and I'll, of course, be available later for questions.